Olá a todos, nós estamos no 8º International Media Plant Meeting, realizado aqui na Exalc, o campus de Piracicaba da Universidade de São Paulo. O tema dessa edição é IA, Inteligência Artificial, na Revolução Científica, Aplicações e Inovações. Diversos profissionais da área estão aqui hoje para se atualizar dessas modernas ferramentas que vêm sendo cada vez mais utilizadas e aplicadas em projetos de genética e melhoramento de plantas. Para conhecer mais sobre esse tema e a experiência de quem está no dia a dia trabalhando com isso, confira agora uma entrevista realizada com um dos palestrantes desse evento organizado pelo GVENCK, o Grupo de Extensão e Genética e Melhoramento de Plantas da Exalc.
Hello everyone, we are here today with Professor Julian Catchen. He's a professor of evolution, ecology, and behavior at the University of Vitória. Hi Julian, thank you so much for accepting our invitation to have you with us. So to start our conversation, could you please tell us a little bit about yourself, your path in science, and how you became a professor at the University of Vitória?
Yeah, thanks for having me. I had an unusual path in science. I started my studies in computer science. At the time, bioinformatics was a much smaller group of people, but my second year of graduate school, I learned about evolution, fell in love with evolution, and soon switched my studies in computer science from networking research to genomics population. So, yeah, many years later.
Yeah. So you are one of the developers of this text software. So can you please tell us about a little bit about the development of the software and what were the challenges that you faced?
Yeah. So we started. Stacks was written when several technologies came together at the same time, place, and so Selecta sequencing, which is now Illumina sequencing, is just getting started. And a new molecular technique called RAD sequencing, which uses restriction enzymes to chop up the genome, was becoming popular. Many versions of restriction enzyme technologies have existed over the years, but this was sort of the newest one. And when Illumina sequencing became available at the University of Oregon, put together Illumina and RAD and got RAD sequencing. So there was no computational way at the time to handle the data, and the lab I was in, I just finished up my PhD dissertation and was getting ready to start a postdoc. At the time, some professors wanted to look at some of this data in the spotted dog, which is an interesting fish. It's an important point in phylogenetic tree. So at the time, Illumina reads were all of 39 bases long. So we asked ourselves, can we sequence? You could only get about 1 million reads at a time, and if we got those reads that were 39 bases long, we put them together in software. And so we played around with some Perl scripts and said, yeah, it should be possible to do. And then we started writing software. Once we actually started getting the results, and built our first very small genetic map in spotted gar, and we saw it started working, and we realized, hey, this could really fill a big gap. And so basically rewrote the software in computer science, came back to me, started engineering the software in a much bigger way. And then it was just really keeping up with the technology for about the next 10 years as the Illumina reads got longer, got more of them. We had to improve our SNP calling capability. Big issue was building software that was handling very detailed molecular data, making it usable by molecular biologists. And that's always been a sticking point where they want to have sort of an oracle where they can say, here's my organism, tell me all the information about it. But in reality, we have to build a system where the scientists can interact with the data and understand what is actually going on in the nucleotide level. That's always been challenging. And a lot of it was learning how to work with the DNA itself. So it's one thing to think about software, but DNA quality is super important in all of these analyses. And scientists want to look at any DNA they get their hands on, whether it's freshly extracted from an animal or whether it's ancient DNA that was found in dirt that's been around 1,000 years. Working with lower quality DNA is always a challenge. And again, learning how to communicate the effects of that to researchers. understand that you can't have perfect information. The DNA that went into the analysis itself was perfect. So sort of learning how to navigate all those things is what's kept us busy.
Yeah, that's a really good story to tell.
Yeah.
So talking about stacks, in terms of adapting like the software to the new technologies like PacBio and nanopore sequencing with founder reads, how is this software going to adapt to these new technologies, and how about the efficiency of the software in processing large reports of data?
Yeah, that's a good question. So I think PacBio and nanopore is excellent technology. We use it a lot in my lab. We mostly use it to assemble pieces. And I think for the next few years, that's really where the strength is. Small numbers of individuals where we can extract high quality DNA, build-fold genomes, even look at RNA, whole transcripts out of the genome. But using PacBio and nanopore directly for population genetics, I think is still not possible. I think we would need several orders of magnitude more volume of that type of data and have it much, much cheaper. But what we can do is take advantage of the huge amount of data the Illumina platform gives us and increase our sample size. So the way we see it is every time the Illumina machine can produce more sequence, we can make bigger sample sizes. So we can look at 500, 1,000, even more than that. Some people are looking at several thousand. But you mentioned that efficiency starts to really matter. We want to process thousands of things. And that's a challenge. We put a lot of work into parallelizing the software. And I think that will have to continue. One challenge is the funding agencies don't typically directly fund software for the sake of software. We've never actually been funded to build stacks. We've always built stacks alongside other research objectives that we were funded to do. And so it'd be amazing to have funding where we could pay professional engineers to come in, really parallelize it. But instead, we basically have myself, my students, a few other people who contribute, and we work on it as we can for get things done. And it's a really fun project, and hopefully we'll be able to, if you asked me 15 years ago, we'd still be talking about software. It's not a chance. So the fact that it's still being used, it's fun, and it gives us, it's a fun project to keep working on and keep improving.
Right. So you're talking about large reports of data, and today in breeding, in animal breeding, generally we use a lot of data from all the places. So from your point of view, what can we expect in terms of the artificial intelligence with new population genetics in the coming years?
I think that's a key question. One of the big questions is everywhere, and as an academic, I constantly think, how can we make sure we're not missing out on these new technologies and how to incorporate them into our research points of view? I think in the population genetics context and in the broader genomics context, AI right now is in a weird position. So if you want to call SNPs using a deep learning algorithm, most of those algorithms today actually visualize what's called a pileup. So they actually convert a SNP call into an image. And that image is specific to the breeds that are aligned in that region. And they then have thousands of those images, which they use to train an image recognition model, which is really a weird way to look at biological data. And several population genetics researchers have done similar. They've turned SNP arrays and populations into images, and they train image models on how to recognize these images. Those are really brittle algorithms that we don't understand. They don't represent true biology. So if you think of something like a transformer model, which is where you see what you see used in this generative AI chat, they take sentences and they break them down into tokens, and then they use these feature vectors where they have all the tokens sort of understand other tokens in context. So you have these strings of words, and there's probabilities associated with those words in the context of the words that surround it. So each token understands what's around it, that gives you the probability. And that's what we need to do in population dynamics. We need to be thinking, what is my feature vector? How do I represent the true, what are the biological characteristics? If I'm talking about things like SNPs, obviously SNP is the token, and then what's around that SNP in the genomic neighborhood or other SNPs in the population. And then using that to train transformative models. I think that's where the field needs to go. So we're a small field. We're not Google. And in fact, Google is the company that has produced the SNP calling variants so far. So we need to sort of reformulate some of those ideas. But again, those companies have large numbers of software engineers that are building models. So we need to find a sustainable way where we could bring more biology to the problem, but be able to do it on a smaller scale where labs and consortiums of labs can work together.
So thanks so much, Professor, for your time, for being here with us to do this interview. Once again, thank you very much. And so don't forget to like and subscribe on our YouTube video. And there will be more interviews in the coming year. Thank you so much. Thank you, Professor.